Alrighty, good morning everybody and welcome to the 2025 Insect and Pest Survey webinar. My name is Riley Rood and I am the Crop Extension Specialist with Saskatchewan Agriculture in Prince Albert. I will be chairing today. We will have CEUs being offered for CCAs on this webinar. In order to get credit, you must attend the entire live event. There will be a QR code at the end of the presentation to collect your credit. If you missed the code and have attended live, please email us afterwards and we can work with you to get your credit. Before we start the formal presentation, I have just a few items to share. Our crop extension specialists put on this webinar series each year. We work out of 10 different offices across the province. Each office contains a wide variety of specialists, and we can help you with any agriculture-based questions. To connect with specialists, please call your local regional office or the Ag Knowledge Center at the number provided here. If you've ever heard the words on the radio, Saskatchewan is 98% completed harvest this week, then you've likely heard of the crop report. Our colleagues out of the Moose Jaw and Outlook office write the crop report each week during the growing season based on data collected from crop reporters across the province. I would first like to thank all of our current crop reporters for helping us achieve this comprehensive report. I would also like to invite any new crop reporters to sign up, so please use this QR code today to sign up. I would also like to mention that the 2025 Crop Diagnostic School will be at Scott, Saskatchewan this year on July 23rd and 24th. More details are coming soon on that event, along with other upcoming field days hosted by local agri-arm research sites. Please note that we will be sharing an upcoming schedule of crops webinars, but in the meantime, check out the invasive species webinars hosted by our agri-environmental specialist team. These will be running at the end of February. Lastly, we want to make sure everyone is aware of the funding available through the Sustainable Canadian Agricultural Partnership. You can reach out to your local agriculture program specialist to find out what funding you may qualify for. So along with all this information I just went through, we have additional resources for everybody under the handout section in the chat box area of the toolbar. So along with this PowerPoint, a document listing valuable resources to be aware of is available. These will also be shared along with the recording after the webinar is complete. Today's presentation will be followed by a live Q&A with Alireza Akhavan and James Tansey. The main hub for today will be your webinar screen, and as mentioned, we will share relevant downloadable handouts with you in the chat box area. The entire session will be recorded, so if you miss anything or wish to revisit any of the presentations, you will be able to. Recording links will be sent out later this week in a follow-up email. An evaluation will pop up at the end of the webinar this morning. Please ensure that you fill out the evaluation, as these help us determine what topics you want to learn more about and help us prioritize research needs for the future. Just a few housekeeping notes in mind for the webinar. If anybody has any questions during any of the presentations, you may type them in the question box at the top right-hand side of your control panel. We will try to get to as many of your questions as possible, and your questions will remain anonymous, only sent to me. During the presentation, you will be muted so that there's no feedback that will interfere with the audio. So today we will start out with Dr. James Tansey. He has been the provincial entomologist with the Ministry of Agriculture since 2018. Prior to this, he worked as a research scientist with the ALK in Post Falls, Idaho, investigating and optimizing industrial production and allergen expression in house dust mites. He came to Idaho from a postdoctoral fellowship at the University of Florida, where he investigated control of the Asian citrus psyllid and other citrus pests. His doctoral research and first postdoc focused on canola entomology in the Dawesdale lab at the University of Alberta, primarily host plant resistance to some major insect pests of canola in novel germplasm. His master's degree examined insect-plant interactions in an important leafy spurge biological control agent. So with that, we will start whenever you're ready, James.
Thanks. Yeah, ready to go. Can you see my screen okay?
Yep, I can see you're good.
Okay. Okay, we'll get started then. Thanks everybody for joining us this morning, and we'll get right into it. We'll have a few slides to cover. So we will be talking about the insect pest surveys as well as Richardson's ground squirrel. So stay tuned for gophers at the end of the presentation. So first and foremost, when we're talking about insects, of course, they're ectotherms. So everything about them physiologically, behaviorally, population growth and population decline are dictated in very large part by the weather. So that is temperature is hugely important, and moist or dry conditions can also be extremely influential. And this varies by species. So we'll talk about some specific cases, but in the spring, of course, it was conspicuously cool and wet, so we had some pretty slow stand establishment, and that had some impact on pest establishment as well, and the impact of some pests. And then warm and dry conditions late season, and that can also have an effect on pests as well. So you can see the cumulative rainfall by the end of May. You can tell it's wet because of all the mud. Yeah, that's the dark brown. Sorry, I never get tired of that joke. Anyway, on to the next slide. But yeah, so again, conspicuously cool and wet in the spring. A lot of discussion last year, certainly about generalist grasshoppers. So we've got four major grasshopper pest species in the province. We'll start from the top. That's clear-winged grasshopper. So this one is, ironically, the markings on the forewings of the clear-winged grasshopper are what are somewhat conspicuous. You can see that camouflage pattern. Not a terribly large grasshopper, about an inch long. Females are larger than males in all grasshopper species. In the case of these guys, very important because they are what are called gramnivores. So that is they really like to feed on cereals and can be devastating pests of cereals. They will feed on other plants, but they really have a preference for grasses. The next one down is migratory grasshopper. This one's considered to be ambivorous. So that is, if it's green and not nailed down, it's going to give it a go. They've got very broad host ranges. They will feed on other insects, alive and dead, and can be devastating pests. Historically, this is the most important pest in the prairie provinces, but this has been overtaken slightly, well, more than slightly, pretty significantly in recent years by the two-striped grasshopper, which we'll talk about last. So migratory grasshopper, as the name might imply, they can fly great distances, and that can be as much as 100 kilometers a day, or that's what's been documented. Packard's grasshopper is a large grasshopper, another ambivore, so that is very broad host range. They have a demonstrated preference for some legumes, but they will feed on a broad assortment of different plants. Really large grasshoppers, so inch and a half long for the females, and tend to have that pale coloration and can have blue hind tibia, which is pretty conspicuous. And finally, the two-striped grasshopper, which is our primary pest in Saskatchewan, and has been for some decades now. So the conspicuous two stripes from between the eyes to the tip of the wings there. You can see the stripes on the forewings, and the black stripe on the hind femur. Those two features are really conspicuous of this species. You can get some different color morphs, and you can see them, be anywhere from this pale cream color to a yellow to greens, and all points in between. So color morphs are pretty common actually in all of these species, but you look at the primary characters on those, and those will help you tell them apart. Uh, in this past year, we had a major emergence of slant-faced grasshoppers in March. Slant-faced grasshoppers are another group of grasshoppers entirely, in a different subfamily than these ones, and can be problematic in rangelands and very numerous in rangeland. So that is, upwards of 70 to 80 per square meter. Not uncommon for them to emerge in March. They are very cold tolerant, and they can come up even while there's snow on the ground. So not uncommon to see these animals come up in March. Typically, these four pest species that I talk about there that are important for field crops, they're going to be coming up early July. And what we saw this year, because of the cool temperatures, their emergence was greatly delayed, and we saw them emerging as late July in some cases. So very late emergence of these animals. They need 100-degree days, base 10. So that is 100-degree days, base 10 Celsius. Uh, Heat accumulated for them to complete development as embryos in the egg and hatch. And we didn't get to that. We didn't get to that spot till mid-July in a lot of places. So emergence greatly delayed. That had an impact on their impact in the spring. But once they did start coming up, they could be pretty numerous in some areas and were damaging. The moist conditions contributed to some fungal outbreaks in these animals. And we saw some pretty broad areas with pretty significant Entomophora grillii infections in these animals, killing nymphs. So typically we'll see the effects of Entomophora in adults, or that's where it's really more conspicuous. And you'll see them climb to the tops of plants. It's called summiting disease. And it obliges its host, this fungus obliges its host, the grasshopper, to, once it's infected, to climb to the top of a plant, grab on for dear life, and then it dies. And because of the nature of its exoskeleton, it's really locked into place. And its innards basically dissolve, and it releases fungal spores on all of its pals. So really a good way for a fungal pathogen to get around. We did have some reports of damage to crops through August, and some accounts of sprays going down. In addition to some very late spring, so well, I mean really July, um, damaging populations of nymphs in a number of different crops, primarily in the Southwest. So this is what things look like in some of the egg beds, and we were really concerned about the possibility of grasshopper populations in the upcoming spring. And had it been warm and dry, as was predicted by a lot of the seasonal forecasts, then I think we'd be having a very different conversation about grasshoppers right now. But you can see, thanks to Carolyn Siemens from SEIC for forwarding this picture to me, all those little flecks that look like rice grains, those are all grasshopper eggs. So very, very numerous in some regions. SEIC conducts our survey, and they typically look at between 1,100 and 1,200 sites per year in the province from July to September. So they're doing an inventory of adults on all those sites. They should have all reached adulthood by late July to early September, and with the warm temperatures that we experienced this late summer, they really sped through development once they did emerge. We had heavy populations detected on more than 50 sites throughout the province. Some reports of damage in central and southwest. And once again, our primary species is two-stripe, so that's Melanoplis bivittatus, and our secondary major pest was Sanguinipes, or the migratory grasshopper. So both of these relatively common, but really two-stripe was outstripping migratory grasshopper, and in some cases by a lot. Here are the results of the 2024 grasshopper survey. You can see relative to last year. Unfortunately, I don't have last year's map, but there was certainly a lot more red on last year's map. What we're looking for next year is an indication of heavy populations coupled with good egg laying conditions, and we certainly saw that this late summer and early fall. Very warm conditions, dry conditions, so very good for egg laying. So we still have some warm spots, leading into some hot spots. So there is still the possibility of an upregulation in grasshopper populations if we have a warm, dry spring. Each female two-striped grasshopper can produce 400 eggs. So you can see there's a real possibility for rapid population increase if you get good survival and good hatch of those eggs. So, you can never keep your guard down for grasshoppers for too long, and especially if it's warm and dry in a number of different regions, definitely be watchful for grasshoppers, despite the decrease in numbers over last year. Another cereal pest that we monitor is the wheat midge, or the orange blossom wheat midge. Thanks to Tyler Wiss for some excellent pictures here. We see an adult, teeny tiny little orange fly, looks very much like a mosquito. So there are other flies, including other orange flies that you can see in wheat crops. The other one that you'll see conspicuously is called a loxanid, and it looks an awful lot like a housefly. So that's what's called a brachycera. It's or a higher fly. The wheat midge is in a different suborder, the Nematocera, or the lower flies, and those are the ones that look like mosquitoes. So they're an older group, a little bit more primitive, and pretty conspicuous in their similarities to body structure of a mosquito. So orange, and it looks like a mosquito, probably looking at wheat midge. Also pictured is the parasitoid, that's Macrogleines penetrans, and that can have a profound effect on wheat midge populations. It is an egg and larval parasitoid, and once again can have a profound effect. Of course, the damage associated with these animals is due to larval feeding and some of the plant defensive responses to that larval feeding. So because of this, we conduct monitoring for it. In addition to that, Ag Canada, led by Tyler Wist, is also doing pheromone trapping, and I'll present some of the data from our provincial survey and the Midge Busters program as well. And you can see there's definitely some overlap, and we're looking at comparing some of these data as well. So no reports of spraying. That comes with an asterisk because more recently I've got some reports of spraying. So for those of you on the call, please, if you're seeing insect damage, reach out to your regional specialist or to myself directly, and let us know. If we don't hear about it, we can't report it and we can't act on it if very little we can do about things that we don't know about. So please reach out if you do have any indications of damaging insect populations. However, reports of spraying were relatively low. With these animals do need 25 millimeters of precipitation by the end of May for normal emergence, and we certainly saw that. But because of the cool, wet conditions, or the cool conditions in particular, once again, they're ectotherms, and their emergence was delayed. And we're thinking right now that there may have been some asynchrony between adult emergence and the susceptibility of the crop. So once the crop reaches anthesis, it's no longer susceptible to egg laying by this animal, and maybe a reason why we had very few reports of spraying. We've seen some good adoption of SM1 resistant germplasm or tolerant germplasm as well, and that's really a very good set it and forget it solution. So if you don't want to be on your knees looking through your crop on Canada Day, then you might want to consider SM1, particularly if you're in a region with elevated wheat midge populations. So here are some of the results from surveys. You can see the lower left, that is our soil core survey, and some pretty good numbers in some areas, Regina, east of Saskatoon, and Estevan area, as well as some western populations. So if you are in those regions, we have seen an upregulation of populations. So it might behoove you to look at SM1. These data are supported by the midge count and you can see here about 38,000 male midges caught. Of course, these are male midges that are caught by the synthetic sex pheromone, and you can also see a pretty good overlap with wet conditions and emergence of the wheat midge. So they really do require moist conditions, but it seems their emergence was delayed because of the cool weather. So one to continue to keep an eye out for. Another cereal pest that's become more prevalent in the last couple of years is Hessian fly. So we've been getting a lot of reports of lodging from really the Battlefords all the way down to the Moose Jaw area, the Moose Jaw Pence area. Pardon me. Some of these are associated with wheat stem sawfly, but we're getting more reports of lodging associated with Hessian fly. And a really quick way to tell that apart is if it's broken off at the first node and you see a plug of frass, then you're looking at wheat stem sawfly. If it's breaking off at the second or third node, you could be looking at Hessian fly. There are a lot of things that can contribute to lodging, of course, and to stem weakness. But if you're seeing these the puparia, or what's called the flaxseed stage, that they look a fair bit like flaxseed and are about the same size, wedged in between those the the stem leaf interface there, then you could be looking at Hessian fly, or you're probably looking at Hessian fly. We're asking that people who do come across Hessian fly, and this is going to be relatively late in the season, late July, early August, when you're seeing this development stage. If you are coming across these, please submit these either to the crop protection lab, and I will get these into containers for rearing. What we're trying to do is rear out the parasitoids, and that top right, that is the one parasitoid that we reared from a number of samples this year. That is a little eulophid. So you look at the size of that puparium, and this parasitoid is about a tenth that size. So it is a tiny little girl, tiny little female beast, and but has a really strong regulating impact on their populations. So there are 4 parasitoid species that have been documented influencing Hessian fly. We're wondering if Hessian fly is currently escaping regulation by its parasitoids. Right now, we have seen an increase in the numbers of sites and an increase in the lodging associated with these. And it's the first time in about 15 years that we've seen this level of damage. So one to keep an eye out for. This is one we're having discussions with our colleagues at the University of Alberta and Alberta and Manitoba about initiating a pheromone monitoring program, and that's looking like a real possibility as going forward this year. So we can have some hard data as far as where populations are occurring. So look forward to that. Another one that we have a formal survey for, and we're entering the second, or we just finished the second year, pardon me, of our formal survey for this, is the wheat stem sawfly. And I mentioned before, where there have been several reports of lodging. Last year's survey indicated really widespread and really heavy populations throughout, a relatively broad swath of the province. The survey concentrated on the Southwest, which is traditionally where this animal is numerous, and this year extended further north and further east. We will be extending the survey further north and further east, particularly in the Regina area in the upcoming year. We did see some heavy cutting, so one field near Pence, 75% cutting, so that is pretty numerous. Several fields, another 4 fields, pardon me, with above 50%, and cutting detected in relatively widespread areas. So average cutting about 11%, so could be pretty significant in some areas. Here are the results of the survey. And you can see we definitely have some hot spots and a fair bit of lodging in the Moose Jaw area. And you can see here that this these animals were quite numerous in those areas. So we're going to be extending primarily in the Regina area the survey further north and further east. So we'll be looking at another probably 20 sites in that area, in addition to the 100 sites that were examined in this past year. And thanks very much to Scott Mears. He is the former entomologist from Alberta, who we've brought on as a contractor to do this work. He knows this animal very well. And thanks very much to Saskweet and SEIC for funding this survey. So tip of the hat to everyone involved. We don't have a formal survey for canola and mustard pests, and this is another one for flea beetles, pardon me, as canola and mustard pests. So this is another one where we're really relying on growers to contact us with information on populations. So yeah, please reach out if you've got damaging populations, particularly populations that are outstripping your seed treatment, and there's looking like there's reduced efficacy of the seed treatment. Please let us know, and I'll in the next slide or two I'll indicate why that might be important. So we got 2 primary pest species. One is Philotreta cruciferae, the crucifer flea beetle. This one typically likes things warm and dry, and we also have the striped flea beetle. This likes things cool and wet. So of course, with the cool, wet weather that we had in the spring, we could see response from these populations and a bit of a shift in local prevalence of these 2 species. It's a wait and see. Where this is important is that the striped flea beetle has demonstrated to be more tolerant of some of the insecticides that are used to control these animals, most notably the group 4s or the neonics. So that is, your Helix and Cruiser. The Buteo Start seems to be very effective for both of these, and has become a pretty popular solution, despite being a more expensive solution for these animals, but is effective for both species. So What we've seen in past years is a shift in species dominance, and that is a move from northern regions to southern regions of the striped flea beetle. That seems to be with drier, warmer conditions, moving these animals further north with greater dominance by crucifer flea beetle. But of course, with the cool, wet weather that we saw in the spring, it's a bit of a wait and see with the upcoming season. So again, wait and see. So yeah, some of the issues that could be associated with dominance of these different species, different climate suitability, and differences in their tolerance to insecticides. So in Saskatchewan, we had significant striped flea beetle pressure in the northwest and northeast. The Spiritwood area was hit really hard by striped flea beetle. Some reports of multiple sprays. Moderate pressure in the rest of the province, primarily associated with crucifer flea beetle, with some hot spots near Kindersley and Swift Current, and significant populations of late season crucifer flea beetle. We don't really have a hard connection between late season populations and spring pressures. They are excellent flyers. They can fly for miles. They don't fly very high, but they can be very long distance flyers. So what you see in the fall doesn't necessarily mean what you're going to see in the spring. So it's a bit of a wait and see. So once again, cool, wet weather prolonged seedling development and stand establishment, and that reduced how quickly these and efficiently these plants were taking up the insecticidal seed treatments. So that was having an impact on efficacy for flea beetles. I was hearing reports about having efficacy for cutworms as well. So Lumiderm and Fortenza, of course, are the products that are associated with cutworm control. Some reduced efficacy associated with that, associated with the reduced ability of those plants to uptake from the seed dressings. So as I indicated before, potential problems with insecticide susceptibility. So to address that, both John Gavlosky in Manitoba and myself have been collecting crucifer flea beetle and submitting them to Dr. Boyd Morey at the University of Alberta to address group 3A or pyrethroid resistance. So DSIS is a really good example of a 3A insecticide. Happily, to date, no indication of group 3A resistance in those populations. However, and this is maybe a worrying trend, as presented at our recent Western Forum meeting, Dr. Boyd Morey and his postdoc have found possible evidence of reduced susceptibility of crucifer flea beetle to the neonics relative to the work that I did in 2007, 2008, which is possibly a worrying trend. Too early to say resistance, but he will continue with that work. But please stay tuned for that. That could be a worrying trend. Diamondback moth is another one that we do a survey for. These are migratory adults, and a bit of an interesting story associated with this one. We were seeing really early, or what we thought was either arrival or emergence of adult moths. So that is, they normally fly from Texas, Florida, or North Mexico on high altitude winds. So they're, you can see this is a kale plant that has been ravaged by diamondback moth from, these brassica veg are grown as winter crops in Texas, Florida, North Mexico, and they have a 12-month field season in those regions, and when it's cooler, they can grow brassica crops. However, they have significant issues with diamondback moth. When these crops are harvested, these animals run out of food, they take flight in large numbers, and they get into high altitude winds. Typically, these are going to be slightly warmer winds, about 300 to 500 meters, although they can get as high as 4 kilometers. They can get to be relatively high. They're just following these winds or maintaining their altitude. The winds themselves are actually pushing them great distances, and they can cover 1,000 kilometers a day. So our traditional thinking with these ones was that they were moving up from these southern reaches and landing en masse, dropping out of the air column en masse, in the prairie provinces or the Midwestern US or as far north as the Yukon. We, because of this early emergence, we collaborated with Ag Canada, Tyler Wist, and the University of Western Ontario to do what's called deuterium ratio analysis. So deuterium is one of the stable isotopes of hydrogen. The other one is protium, and they occur in stable ratios and in water. So hydrogen, of course, gets incorporated into H2O water. That gets incorporated into plants. Insects eat those plants, and so you can get an idea which part of North America these animals came from by the ratio of protium to deuterium. And so this work was pioneered in large part at the University of Western Ontario, so we submitted samples to them. And the results were, happily, these animals are not overwintering here, which is what we were worried about with very fresh-looking adults, because they can be important pests of seedling canola, and if they are emerging here and overwintering here in some numbers, that could really change our approach to these animals. Happily, they are not overwintering here, but some other information that came from this is that what we thought was a point-to-point migration turns out may in fact, or there's strong evidence for that because of sources from the Midwest US. We may be looking at a generational migration, a little bit like you see with monarch butterfly. So some pretty interesting findings from this, but I think the most important is that they are not overwintering on the prairie provinces in large numbers. So this is what monitoring looks like. You may or may not have seen these Delta traps, and thanks to those on the call who participate in our monitoring programs. These are synthetic sex pheromone traps, so we're taking what males think are going to be, a terrific day and turning them into a very bad day. He thinks he's finding a female. He is in fact finding a glue board. So yeah, bad day for him. We had 59 traps throughout the province. Catches were moderate. Happily, we didn't see large populations and few reports of spraying. So looking at this as a map, we have this is the results from the 2024 survey. You can see here, not a lot to write home about. We had some moderate populations in the Northwest, and in some other areas, but for the most part, populations were very low. Another one that we continue to monitor for is Bertha armyworm. This is a native species, and once again, cool conditions had an impact on these animals. So we saw peak emergence late July. Typically, this is going to be early July to mid-July, so at least 2 to 3 weeks later than we might normally see. Our normal advice for these ones is start monitoring 2 weeks after peak emergence. And really, I mean, we were looking at some very late crops by the time we saw the normal advice for monitoring for these for larvae. So we still continue to monitor for these 292 sites monitored. No spraying reported for this one. We had some reports of misidentification of clover cutworm as Bertha, and some spray going down for that one. Typically, consider the thresholds to be comparable, and the control products that are registered for Bertha will be effective for clover cutworm as well. So some moderate populations. However, we'll take a look at the map, the graph, and once again, this is a synthetic sex pheromone lure. So taking what males think are going to be very good days and turning them into very bad days, since decades now. So you can see the trap catches remain low. We're going to continue to keep an eye out for this one. This is an animal that typically outbreaks every 6 to 8 years, and is regulated in large part by natural enemies like viruses. So we're getting to be due for an outbreak. So we will continue to monitor for that. Stay tuned to the ministry website and to the Prairie Pest Monitoring Network, as well as social media accounts for our regional specialists and other ministry personnel. If we're seeing an outbreak, we'll try to get the word out as early as we can. We saw increases in numbers of cabbage seed pod weevil in our survey this year. Both adults and larvae of this animal can be damaging. You can see an adult pictured here. At the end of her rostrum, she has the more typical insect feeding parts, so mandibles, maxillae, etc. And she's going to use those to chew a hole into a developing pod, turn around, lay an individual egg in it, and mark it with what's called an oviposition deterring pheromone. So what she's doing is labeling that pod as no vacancy, so that another female doesn't come along, lay an egg in that pod, and possibly compete with that larva. They're cannibalistic if they encounter each other inside pods, so it's very rare to see more than one larva. That larva is going to consume 3 to 5 seeds, chew its way out. So in addition to the seeds being consumed and the potential for bud blasting by adults, There's also those avenues both from the entrance hole and the exit hole for pathogens to get in, and it can contribute to shatter. So these a number of different mechanisms for this particular insect to damage canola and mustard crops. Some sprays going down for this, and we saw significant elevation of populations in the Swift Current region with some economic populations. So taking a look at the survey data, you can see, yeah, it's Swift Current and South and west, elevated populations, as well as south and west of Regina, as well as someone indicate if I'm still broadcasting.
Can you try that again there, James? We're just having a little bit of troubles.
Yeah, okay. Can you hear and see everything now, or
I can hear you now.
can you see the presentation?
Yep.
Okay, excellent. We'll keep going then. So yeah, you can see here some elevated numbers of cabbage seed pod weevil, and some sprays went down. Happily, a lot of these areas with economic damage, we managed to get the word out to the growers in a pretty timely manner, and they managed to get sprays down and control these populations. So keep an eye out for that one, particularly if it's warm and dry. We continue to survey for the pea leaf weevil. Adults can be damaging. You can see the conspicuous notch feeding on plants that can cause damage, but typically it's going to be the larvae tunneling into the rhizobian nodules that are going to cause the most damage, and that can result in a nearly 30% yield hit. And we've seen some pretty significant damage from these animals last year, particularly in northern and eastern sites right down into the southeast. So we increased our survey geography further north and east, and increased the numbers of sites this past year. So long and the short of it, I got a lot of quality time in a four-wheel drive, bouncing along in the forest in the north, which was actually quite a lot of fun. So you can see why we were concerned. You can see, elevated populations in 2023. We had a near collapse of these populations in recent years, to the point where we were actually not considering this animal a pest. You can see it has upregulated, and we're going to continue the survey. Yeah, populations ebb, populations flow. So we're going to continue to watch for it. Happily, we didn't see the dramatic numbers that we saw last year. We did see some numbers in some fields, but overall numbers were very low throughout the province this year. I did some modeling a few years ago that indicated there was a strong connection between snow cover and the success of these animals overwintering. And we didn't have much in the way of snow cover in 2023-24, and I think this had a pretty profound effect on their populations. They are European, so they're not terribly cold hardy despite being invasive. And their supercooling temperature in, about that -8 region. So they're not terribly cold hardy, not as cold hardy as some animals that evolved on the Canadian prairies. So if they get the snow cover like we're seeing this year, of course, there could be the possibility for those populations to upregulate again. What we advise for control of these insects is seed treatments with neonicotinoids. The evidence for sprays having a meaningful effect is pretty limited. So yeah, seed treatments in areas with elevated populations. Of course, we've seen decreased populations in the past year, but with the snow cover, we could see those bounce back. So not a survey, but we did have some reports of poor performance of lambda cyhalothrin reported for lentils or p-aphid in lentils. So that is the active ingredient in Silencer and Matador. We initiated a field testing kit in collaboration with Ag Canada and the University of Saskatchewan, as well as Saskatchewan Pulse Growers. And thank you to sincere thanks to all our collaborators here. You can see Faith Hilson, our intrepid summer student, still smiling after putting kits together. So really what we were doing, these were vials that were coated on the inside with either full rate, half rate, quarter rate, or a water control, 4 replicates each. We would ship these out to sites, and really what you do, so just to backtrack a little bit, you put the insecticide in, you put the vials on a hot dog roller. Yes, a hot dog roller. We have a pretty interesting lab setup now with a hot dog roller. Haven't used it for hot dogs yet, but hot dog season is coming. Um, Put them on the hot dog roller and roll them until the insecticide evaporates. Obviously, you do that in a fume hood. And once that insecticide is dry, you can ship those out, introduce 10 aphids, look at the level of mortality. So we can see that we had relatively broad reduced efficacy of lambda-cyhalothrin. We use silencer in this case. And in one case, we had 75% survival exposed to this insecticide. So we're really not certain if this animal overwinters in large numbers on the prairies. It seems likely they do. So this is a trait we're concerned has become heritable in our aphid population, and we will continue to keep an eye out for. So we're going to have kits in our back pocket for the upcoming field season. And please reach out if you have concerns about pea aphids. Another one that we had a side of table survey for was true armyworm. We had some sites in the Craven area, some sites in the Saskatoon area, but primarily in northern seed forage growing areas, and some good catch numbers, moderate to good catch numbers up there, and at least one indication of damaging population in crested wheatgrass. We're discussing whether we're going to go forward with that again this year. I'm hoping we do. This is an animal that I think can be pretty important. One that we don't do a survey for, and we actually don't have the capacity or tools to do a survey for, is rice worm in wild rice. This is one that is harvested by First Nations community in northern regions. So it is organic production and in fish habitat. So this really limits the number of control options that are available for this insect to basically nothing. There are no commercially available products available to monitor this animal, so that is no synthetic sex pheromones are available to my knowledge. If somebody knows of one, please contact me. So we don't really know what to expect from this animal in the coming season, but it was very damaging in those populations this past season. Another one that we do not do a survey for, but certainly keep an eye out for, are Delia species. We've got multiple Delia species, primarily Delia radicum, the cabbage root maggot, and it can be very damaging to brassica veg. Of course, the outlook area is surrounded by canola, which is an alternate host. Not terribly damaging currently with modern varieties to canola, but we do still monitor for it as part of the disease survey in canola. So numbers were slightly elevated this past year, and we're going to continue to keep an eye out for this one as part of the canola disease survey. So there is currently an emergency use registration for Cymegra. That is a group 30, so a relatively new mode of action for insecticides. So interesting to see new modes of action for insecticides. It is highly toxic to pollinators, but not systemic. And because of the timing of application and the fact that it isn't systemic, we feel it doesn't represent a risk to pollinators. Another one, and this is the last insect that I'll be talking about, is spotted wing Drosophila. Sharp has our provincial fruit specialist has really taken the lead on this one and done an excellent job monitoring a number of different sites with Sentry lures. So there is a commercial monitoring product available for this called Sentry, SCENTRY. If you're curious, they have a website, and this product is available for purchase. If it ends in berry, this animal will attack it. It lays its eggs. You can see well, it might be difficult to see in this photo. The female has a saw-shaped ovipositor that she uses to pierce the skin of healthy developing fruit, lays an egg in that, and this really ruins the quality of the fruit. So they will attack a broad assortment of different bush berries, cane berries, tree fruit, and tomatoes, and can be a real problem for our fruit industry. So a lot of monitoring going on, a lot of control products with emergency use registration and otherwise registered for its control. So we will continue to keep an eye out for this one, broadly distributed and quite numerous throughout the province. So some other surveys. So yeah, sorry, I fibbed. Not the last insect I'll talk about. These are the last insects I'll talk about. Swede midge. This is an AFC led program that we contribute to. No Swede midge caught to date, which is excellent news. This can be a significant pest of canola and mustard production in Ontario, and we're happy they haven't arrived yet. Japanese beetle, limited number of traps, but this is one that we collaborate with CFIA and Ag Canada on. Pardon me, in Saskatchewan, again, a limited number of traps, but happily none caught to date. Very broad host range for this one, primarily a fruit and turf pest. And yeah, the gopher bug. So moving on to vertebrates. This is Richardson's ground squirrel, as I'm sure many of you are aware. It is semi-fossorial and semi-social, so it spends part of its time underground, and it only conditionally likes each other, and they only conditionally like each other. Their colonies are based on female kin groups, so that is mother-daughter, mother-daughters, grandmother, daughters, grandmother, mother, daughters, female cousins, that sort of thing. They typically drive males out to the periphery of these colonies. Males only gain access for a short period of time. So female estrus, or when they're receptive to males, is only 4 hours per year. So I guess you can see why the males are really fighting pretty hard to gain access to the females. They are native animals, native to the American shortgrass prairie. And can cause significant damage in some crops and did conspicuous damage in some regions. You can see a field here. This is or was canola seedlings. This is after a second planting, and this grower put down literally tens of thousands of dollars of control to try to limit the damage of these populations. I suspect he probably had an effect, but it may have been a bit late to get the control down. So early is really important. And you can see here they work well into the field, which is conspicuous. Another conspicuous thing you may notice is that the cereal crop right next door is fundamentally undamaged. So they really have a predilection for canola seedlings and can be quite damaging. I'd never seen them move this far into a field before. So some very conspicuous damage. So for control, if you're going to use poisons or if you're going to use blunderbuss or some other trapping, drowning, some other factor, the best time to do that is March 15 to April 30. If you see a large activity of migrating bald eagles in a field, they're homing in on the emerging males. So the adult males are coming up first. They're going to feed on their caches and jockey for position to access females. Females are going to come up a couple of weeks later, and and these will of course give birth to the juvenile males and females. They'll tolerate the females more than they'll tolerate the males. They tend to drive the males out earlier, both adults and juveniles. So if you're driving down a country road or a highway and you get that conspicuous tup tup, you've probably just met a male. So some discussion about the efficacy of some of the alternatives that are currently registered for strychnine. We did some work in 2022 and 2023, and they all had similar results. So just to encapsulate this, these are the results that we had in Saskatchewan. We collaborated with the Ministry of Agriculture in Alberta on this. So we had sites in Alberta, sites in Saskatchewan, replicated field trial. And these are some of the results that we came up with, was that the zinc phosphide baits were as efficacious as strychnine. If used early in the season, you fire them down the hole, and they can be very efficacious. We had some reduced efficacy in this case with some of the anticoagulants. Wasn't the case in both years, wasn't the case on all sites. But as a rule, the Rosol was a less efficacious performer than some of the other ones, and that is an anticoagulant. And I suspect the reason why was the time it takes to kill. So we left this experiment run for about a week, and it can take that long to start to see major mortality associated with Rosol. In addition, you may be required to put down multiple feedings, both for Ramet Green and Rosol, whereas with the Burrow oat bait or ZP rodent oat bait, the zinc phosphide treatments, that's a single feeding. So very important to apply baits before green up. Do that as early as late February in some regions. Really the ones you want to target are those reproductive females. It doesn't take very many males to contribute to the next generation. Each male can mate with 10 females. Look at the zinc phosphide products, and again, early application is important. Consider the anticoagulants; they are still good products, but also consider that fresh plant material. So this is where green up is important. Vitamin K is an antidote for these anticoagulants. Actually, mess with uh, the coagulant effect of vitamin K. So vitamin K is a precursor to coagulants. So as an anticoagulant, of course, vitamin K becomes the antidote for that. Also look at the Saskatchewan Gopher Control Program. Uh, it is a program where you can claim 50% of your costs for registered gopher control products, which can be important. And you can also claim a 50% rebate on non-chemical controls like Raptor platforms. And this is an example of one that we built in the Craven area. We're still waiting for it to be occupied. We got it up kind of late. But this area is dominated by northern harriers, so we're hoping that they establish there and have an impact on the gopher populations. Also, have some tolerance for predators. Badgers, in this case, can have a significant effect on Richardson's ground squirrel population. Yes, I know they can be damaging by themselves, but if you can be tolerant of them in your region, they can have an impact both this year, and if there are no gophers to populate for next year, it can be an ongoing control. Short-tailed weasels. This was a photo from my place actually of a short-tailed weasel stealing babies from mom as they were suckling underground. So this caused quite a bit of commotion in the old gopher colony. A lot of yelly gophers. So we had some questions about the level of grasshopper, pardon me, of gopher populations, and you can see here that both the claims and the indemnity paid are lower than when strychnine was registered. So I don't know that this would be really good ammunition for petitioning for the re-registration of strychnine, and we have a bit of a long history with PMRA. The likelihood of re-registration of strychnine I would consider to be very low. So please consider alternatives for strychnine. And as indicated here, we saw localized populations and some pretty heavy damage in local areas, but over the entire province, populations are down relative to when strychnine was used. We do conduct an online Richardson's ground squirrel survey, and you've got 2 days to participate left to go. So we're looking for an indication of damage severity, acres affected, control inflicted, natural enemies, influential events, so that influential weather events, and takes about 5 minutes to do. Point your photo at this QR code, and I'll leave it up for a moment. But it's really invaluable data. We had 174 respondents last year. You don't necessarily have to have had issues with Richardson's ground squirrel. So if you've had no problems with Richardson's ground squirrel, we want to hear about that too. So zeros are still data, and we want to look at what was going on in your region with natural enemies, with past control, with weather events, and try to tie A to B and try to figure out what's going on with the weather with these animals, and try to give some predictive power. So prior accounts, publications have indicated that warm, dry springs influence gopher populations. Of course, with the very cool wet spring that we saw this year, we shouldn't have seen damaging populations. If those models were the whole truth, or were an accurate representation of everything that was going on. So we're looking for more information so that we can get some more predictive power and feed that into our model. And with that, hopefully everybody's had the time to point their phone at the QR code and participate in the gopher survey. Oh, just one last slide. A gopher is not a gopher is not a gopher. So gopher, gopher, gopher. So what they are called on the prairies, gopher, Richardson's ground squirrel, also Franklin's ground squirrel, also 13-line ground squirrel. The only ones that are not called gophers are the gophers, the northern pocket gopher, which are called moles, and we don't have moles here. That's actually the star-nosed mole, which actually occurs in eastern, southeastern Manitoba. It's an insectivore. That's the cute, peculiar little beast you see held up there. Yeah, they're tunneling through the soil looking for insects, and of course the often mistaken Sasquatch. And finally, with that, I will take any questions. Also, if you have any questions or comments, please reach out to me either directly through email or on Blue Sky. That's my social media account. And with that, I will address any questions.
Thank you for the great presentation, James.
Thank you.
Just a reminder, if anyone has any questions, please type them into the chat box there. Um, Another note on the ground squirrel survey there, we will send the link out with the follow-up email.
Excellent.
Alrighty, so first question I have, do Entomothera fungal spores overwinter, James?
The thinking is yes. It's actually it's a super interesting system. There are definitely people with considerably more knowledge about Entomothera than I have, but there are different isolines. So that is, one isoline can have a really profound. So consider these to be races of Entomothera grilli. One can have a really profound effect on two-stripe grasshopper, and clearwing grasshopper occurring in the same field is not being affected at all, and vice versa. So these different isolates can have different effects on different species. Another really interesting paper that came out is there is an impact of formicine ants. There's one formica ant, a little black ant, that will actually carry spores to grasshoppers. What the adaptive significance of this, why an ant would do this, is beyond my understanding. But there is evidence that ants will actually carry these to grasshoppers and stick them to grasshoppers. Why this occurs, I don't know. But as far as overwintering, yes, my understanding is they will overwinter as spores.
Alrighty. And also, James, could you turn your camera on if it's not already? Next up, I have a comment. One of the audience members had commented that near Nokomis, they were picking up 400 Bertha army worms in their traps, and that they should have sprayed as they seen some damage.
They saw
Um,
400. Okay, yeah, we did have some moderate numbers in traps. Yeah, I seem to recall some 300 plus numbers. And they did see some damage, at threshold.
I'm not sure if I have that comment, but yeah, just a good plug for pest surveys too to join up to be a crop reporter. Like the
Of course,
more
yeah,
people we have on that, the better.
yeah, no, and to that, yeah, please, let us know during the course of the season. I'm going to amend my report to include that. So yeah, thanks for the information.
Next up, do we have any information on the brown marmorated stink bug or Japanese lily beetle? I haven't heard of
Lily,
it.
go ahead.
No, I was gonna say they hadn't heard of it in Sask, but they were just wondering if there's any up-to-date info on that.
Okay, just for clarity's sake, so Japanese beetle we do monitor for. We have yet to detect it, and they have actually achieved local eradication on the lower mainland in British Columbia. They're continuing to monitor for it, but their control efforts have been very effective in British Columbia. Happily, we haven't seen it in Alberta. So, lily leaf beetle, I think there may have been a little bit of crossover there. We have had numerous reports of lily leaf beetle both in Saskatoon and in Regina. And we Sydney Worthy, the former City of Saskatoon entomologist, and I looked pretty hard at trying to get some of the Pteroscus parasitoids. They are very effective biological controls for lily leaf beetle. They are so effective, in fact, that they eat themselves out of house and home, and they cannot be recovered where they're released. So it's yeah, they actually decimate the population of lily leaf beetle. So we contacted both Cabby Biosci in Switzerland, which was the original source of these beetles. They no longer have them or access to them. They don't currently have a colony. There was a colony that was led by a researcher by the name of Cappuccino. She has since retired. The colony no longer exists. It seems everyone who was associated with that project is now retired, and gaining access to that particular parasitoid was impossible this past year. We're going to keep our ears open for this upcoming season. There were releases done at Olds College. Once again, they ate themselves out of house and home, and releases done in the city of Calgary. So we'll reach out to them. If there's any indication that we've got established populations of Pterosticus, we will reach out to our colleagues there and see what we can do about getting them released in Saskatchewan. And what was the third insect?
The other one there was the brown marmorated stink bug.
Yeah, we continue to monitor for that. And for those of you who may not be familiar, I don't have a copy of the. Let me see if I can call it up here and share it on screen. Uh, oh, yeah, you probably. Am I sharing my screen right now?
Yep,
Let
I can
me
see your.
let me unshare my screen as I go picking through my files. So there are a number of invasive species that we are keeping an eye out for, including brown marmorated stink bug. There's also a yellow spotted stink bug that can actually hybridize with brown marmorated stink bug. So I guess that makes yellow spotted brown marmorated stink bug as the hybrid. Yeah, how's that for a mouthful? Um, Sorry, I'm just trying to call up this poster. So I do sit on a national committee along with Megan Van Koski from Ag Canada and John Gavlosky from Alberta, in addition to a number of other folks. And we have put together a poster for different regions of the country. And let me see if I can't share this.
We can also share that poster along with the email if you can't find it there, James.
Yeah, I've got it open right now. I've got the spinning circle of computer confusion right now. Sorry, pardon the delay. Okay, it should be doing computery things.
Perfect. Yeah,
There
we
we
can
go.
see it.
Okay, so this I will share. You can see yellow spotted stink bug. Of course, we're concerned about spotted lanternfly as well. We have some regulated pests on this, so there is actually an obligation to report. And we're looking at an online information gathering reporting system that anyone can tie into for the upcoming field season. So, yeah, we've got a fair number of them here. And so once again, yeah, we can share. And you see Japanese beetle here. So some tree pests, some crop pests, some fruit pests, and of course the northern giant hornet, or what was formerly called the Japanese hornet, and what was taken up by the press as the murder hornet. Um, yeah, the problem with common names. So I guess, yeah, with that, I guess maybe we'll forward this in the email.
No, that's great info. Yeah, we'll include that in the email there. The next question I have is Saskatoon's trees, especially American elm, were severely impacted by, I assume, the armyworm. Are there any reports regarding this pest?
Yeah, there was a report actually, and it's cankerworm. Yeah, so it would be spring cankerworm. I'm going to defer to the city of Saskatoon entomologist, and I can, if you will forward your contact information, I can forward you on to her. Yeah, she just started a short time ago, but presented an excellent report at our Saskatchewan Entomological Society. Oh no, it was a different meeting, as a Saskatchewan Insect Management Committee meeting most recently. So she did present a full report on tree pests and other urban insect issues in Saskatoon. But yeah, it was spring cankerworm that was the major issue, and could be quite damaging. There were also Dutch elm disease reports, and a fairly large number of them both in Saskatoon and Regina.
Okay, good to know. Next question. Is there any dormancy in grasshopper eggs? Is it possible for unhatched eggs from 2024 to contribute to the 2025 population, or would we expect all eggs hatched and the 2025 population to be exclusively based on the last year's adults?
That is an excellent question. No, I love questions like this, and the answer is yes. If they encounter very dry conditions, then embryonic development will stop. Yeah, so that is they need a certain amount of moisture, and if that egg falls below about 13% moisture, the embryo will actually die. As a way to sort of preserve that or prevent that, the embryo will actually stop development at a certain time, but that's only under dry conditions. Of course, we had very wet conditions in a lot of parts of the province, and there's no mechanism for what it's called is delayed diapause. And a lot of insects will engage in this, and they can, wheat midge is another example where under very dry conditions they will engage in what's called delayed diapause and actually stop larval development, and just hunker down, and they can hunker down for multiple years. Same thing for grasshopper eggs, but with the wet conditions, there's no mechanism that I'm aware of for them to engage in that delayed diapause. So I suspect, really what you see is what you get for next year, or for this coming season. But yeah, if conditions had been warm and dry, then yeah, for sure you can see delayed diapause.
That's interesting. I didn't know that. That's cool. Another question. With the resistance that has been noted in aphids, what should producers keep in mind when managing aphid populations?
Yeah, I think look at alternate products. There are multiple products registered for aphid control in pulse crops. I mean, a real good fallback for a number of growers. I realize that the lambda products are cheap and plentiful and can be very efficacious when they're very efficacious. But carbine is a very good product. I'd have to open the guide to remember the list of products, but they also include, and asterisks here, I may be remembering wrong, Savantoprime and Movento as well. So there are other products that are not group threes that can be very efficacious for aphids. Where those come in economically, I can't comment. I know carbine is a little more expensive than silencer. I think silencer was coming in at about $7, $8 an acre, and I think carbine was coming in 10 or $11 an acre. That's completely anecdotal though. So
Okay. We just have a couple more questions. Are the are all other gophers aside from Richardson ground squirrels pests as well?
conditionally, yeah, yeah, conditionally, they're all labeled on most of the control products. But the only place I've ever seen Franklin's to be a problem is on woodland, grassland margins. And the only place I've ever seen them be a real pain was in a graveyard. So typically what you're going to see, in an agricultural setting is going to be Richardson's ground squirrel. As you get further north, particularly as you get much further north, then you can see different burrowing rodent species. But 13-line ground squirrel is very rarely a pest. They're not colonial. They really don't like each other's company all that much unless it's mating time. You can see them grouping together loosely if they've got some soil that they like, or there's ample food in the area. But it's pretty unusual to see them as causing economic damage to crops, pasture, range, or hay crops. Typically, the problem is going to be Richardson's ground squirrel. I should probably mention too, northern pocket gopher can be damaging. It's a bit of a conspicuous root feeder, so it can be damaging. Of course, the burrows can be a problem for equipment. Um, they're not colonial either. They really don't like each other's company very much, and they're active all year. So under the snow, they're still active. They spend all their time underground. They're going to emerge for short periods, for short forays to find food. And you can see it's going to be about the size of, silver dollar. For those of you of a certain vintage who've seen a silver dollar. But yeah, the opening is, bigger than a toonie. These are feeding holes. They're blind holes, so there's not going to be that tip or that pile of soil anywhere near those. And those are feeding holes. They're going to dart out of those, to feed on plants. They can be damaging, but on the upside of that is because they're not colonial, all of those mounds in an area are probably associated with one pocket gopher. So if you have one of those tips or one of those mounds, steel pole and probe about 24 inches, 2 feet away from that tip, in a circle, and you'll probably find the main tunnel. If you find the main tunnel, this on the condition that you actually have a damaging gopher. I mean, presence isn't necessarily damage, and they're important nutrient cyclers. I mean, they're good for moving subsurface surface soil to the surface. That's pretty obvious, I think. But if you do have a problem with on gardens, that sort of thing, probe the soil, and if you hit a soft spot, that's going to be the main tunnel. Put a trap in there. Physical traps work really well for these animals, and that can remove that animal from the equation. Uh, there's some recommendations for putting poison bait down the feeder holes, but they're pretty active diggers. That may be effective, but it's not as effective as putting a trap in the main tunnel.
Hmm. No, that's some good information. I got one more question for you, James.
Sure. Yeah.
Do you have any suggestions for areas seeing cricket damage in crops? They're referencing the west central area, I think.
Yeah, no, we've seen elevated cricket populations in the last 2 years, like I've never seen before. And I think that's, anecdotally, but that seems to be the account. Unfortunately, we don't survey for them, so we don't really have a hard look at, where they occur. We only have so many resources, and we need to concentrate on some important economic pests. But yeah, we have had a lot of reports of elevated cricket populations in a number of different crops. I think most conspicuously in canola crops, munching on pods, and there's some thinking that they may be secondary feeding. Flea beetle populations come in late season, do a little bit of stripping of those pods, and then the crickets are stimulated to continue feeding. That's speculation at this point, but they are strict. Well, they're strict omnivores. How do you like that? They're broad omnivores. So yeah, again, lots of different plants. They will happily feed on other insects. I've got some photos of multiple field cricket tackling a live grasshopper and consuming it. So yeah, they will feast on other insects. Again, if it isn't nailed down, they will try to use it as a food source. As far as control goes, there is nothing specifically registered for their control. However, and this, comes with a caveat. You have to consider that there is nothing for registered for their control. They are relatively closely related to grasshoppers, and products that are registered for grasshopper control should be efficacious for them. But I can't advocate for their use because they're not registered for that use. How's that for a non-answer answer?
No, that sounds good. I think that's all the time we have. Thank you, James, for all the great information. We appreciate the time you took here for us today.
Oh, no, my pleasure. Absolutely.
So next up, we will have Dr. Alireza Akhavan. Alireza is a provincial plant disease specialist at the Saskatchewan Ministry of Agriculture with a master's and PhD in plant pathology. He focuses on biosecurity and disease management to enhance crop health and productivity, developing guidelines to prevent disease spread and leading initiatives like the Saskatchewan clubroot and verticillium monitoring programs. Previously, he taught at the University of Alberta and worked with Agriculture and Agri-Food Canada, earning multiple awards, including the Agriculture Teaching Hall of Fame Award. Dr. Akhavan is actively involved in research and extension activities, providing valuable insights and support to farmers and agronomists across Saskatchewan. We will start whenever you're ready, Alariza.
So can you just confirm that I am in presenter mode?
Yep, looks good for me. I can see you. I can see your slides.
Okay, that's great. So good morning, everyone, and thank you, Riley, for the introduction, and thank you all for joining us today. I will be sharing today some of the key findings from the 2024 Saskatchewan Pulse and Cannula Disease Survey surveys, as well as high-level insights from the Fusarium head blight survey for wheat and barley in province. So the work, as it sounds, the work that I am presenting today is a teamwork involving like multiple organizations and many people. So I would like to acknowledge the invaluable contributions of the provincial specialists, crops extension specialists, and the crop protection laboratory from the Saskatchewan Ministry of Agriculture, as well as the SARM plant health technical advisors from the Saskatchewan Association of Rural Municipalities, Sask Oilseeds for their partnership in running all three canola disease monitoring programs we have here now in province, SCIC for sure, and Discovery Seed Laboratory, and the surveyors from industry like Meadow Lake Co-op, Bayer Crop Science, Nutrient, Canola Council of Canada, as well as some of the volunteer agrologists and producers who help us running these programs. So let's start with 2024 pulse disease surveys. In 2024, we looked at 55 lentil crops, and we surveyed for foliar and root rot diseases in those crops, and 40 fields for field pea, and we looked at the same for foliar and root rot diseases. And for very first time province, we had a quite small scale, but new survey for dry beans, and we looked at foliar and root rot diseases in this crop as well. So starting from lentil, a total of 55 lentil fields we surveyed in province in 2024, and the survey was conducted between July 12 and August 8. In each field, 10 plants were sampled from each of 5 sites arranged in a W-shaped pattern, so 50 plants in total per field. Each site was located at least 50 meters from the field edge and at least 30 meters from one another. So for lentil crops were assessed for prevalence and incidence of anthracnose, ascochyta blight, sclerotinia stem and pod rot, botrytis stem and pod rot, stemphyllium blight, and root rot complex. So again, because I will use the three terms very frequently, it would be prevalence, incidence, and severity. Perhaps I just remind you that incidence is calculated as the percentage of plants assessed, like out of 100 plants or 50 plants total per crop, how many had symptoms of a certain disease. This would be incidence. So, so it is actually the number of plants with disease symptoms, while prevalence is a measure of the presence or absence of the disease in the field. Severity is the proportion of plant with disease. All of the disease assessments were made mainly based on visual symptoms observed in the field by trained surveyors, and samples were tested in the lab, most cases only if requested by an individual surveyor for further confirmation. So for lentil, again, anthracnose was the most prevalent foliar disease in 2024 and was identified in 85% of the surveyed crops with an average incidence of 38%. With an average incidence of 38% and with an average severity of 0.6 out of a 0 to 5 disease severity scale. These numbers, all of them, all three, are higher than last year that we had not only, but we had 74% of lentil crops with anthracnose, and average incidence was about 20% and severity was 0.3. So we had significantly more anthracnose seen this year compared to last year. So moving to another relatively important lentil foliar disease, it is Stemphylium blight. We observe it in 25% of crops again this year with an average incidence of 4%. which was quite similar to what was observed last year in 2023. Moving to Sclerotinia stem and pod rot, we observed it. We observed it in 44%. We observed it in 4% of crops with an average incidence of 0.1. So not a big player among diseases. Sclerotinia stem and pod rot were found only in 2 fields, each of the east and west central regions. So moving to Ascochyta blight, which is quite rare to find these years, recently with typical symptoms, but suspect symptoms suggesting Ascochyta blight was observed in 4 fields in southwest and 1 field in west central regions with a low average incidence of 0.9%. However, especially for ascochyta blight, it's important to note that diagnosis was only based on visual symptoms in the field, and plants who had visual symptoms that were consistent with ascochyta blight were not further investigated with additional testing in the lab. So overall, I can say for ascochyta blight this year and last year was quite rare to find with typical symptoms. However, root rot complex is going to be a big significant issue for lentil. Again, we observed it in 85% of the surveyed crops this year, and the average incidence was quite high at 41%. A high level of root rot was observed across Saskatchewan, and these numbers for the prevalence and incidence are higher than those in 2023, and also for the severity is slightly higher than 2023. So overall, we saw more root rot in lentil crops this year than last year. So as you see in this regional breakdown table for root rot complex and anthracnose, these two diseases were prevalent in all regions of the province, while for other diseases we may have had more prevalence in certain regions. If you want to further learn on regional breakdown of the disease, all the reports are available online on the Western Forum on Pest Management website, and I can click on this link and this is the website. So this is the Western Forum on Pest Management. Of course, it is not only for diseases; it is also for crop pests. So if you get to this website, Western Forum on Pest Management, you will see on your left-hand side that there are two boxes. Western Committee on Crop Pests that has the annual provincial insect reports. But if you scroll down to the bottom portion, you will see the box for Western Committee on Plant Diseases. And if you look at the annual disease situation reports, you click on it, you will see reports back to 2015 reported there. And if you just open this window, you will see all the reports are there for Western Canada for almost all the crops, cereals, and pulses, and so on and so forth. So if you are interested to kind of learn more on the regional breakdown for all the crops, perhaps you can review those reports. The same for incidence. So the other table was for prevalence, but same pattern was also observed for incidence of the disease, similar to what I just showed for prevalence. So high incidence of root rot and anthracnose were also seen across the regions, while the incidence reported for other diseases varied among different regions. So moving to field pea diseases, in 2024, we surveyed 40 field pea crops across Saskatchewan, and the survey was conducted between July 9 and August 6. For each field, we sampled, like lentil, 10 plants from 5 different sites arranged in a W-shaped pattern, ensuring that each sampling site was at least 30 meters apart. And in each sampling site, we looked at 10 plants, so 5 times 10, 50 plants in total per crop. Crops were assessed for the prevalence and incidence of root rot complex, Mycosphaerella slash Ascochyta blight complex, which are actually three different pathosystems that make that complex. Then also for downy mildew, white mold, bacterial blight. In addition to prevalence and incidence, we also calculated the severity for the root rot complex and Mycosphaerella ascochyta blight complex. And again, for most part, the disease assessments are made based on visual symptoms in the field. But if a surveyor asks for further confirmation, then they can send samples to the lab and lab will complete the analysis. So Mycosphaerella slash Ascochyta complex was present in almost every field. 95% of the surveyed fields had that disease. Average incidence was also quite high at 61%, but average severity was quite low at 1.8 across the province. And given that specific disease severity scale is a 1 to 7, so it's not 0 to 7, it's 1 to 7 established that way. So 1 is a healthy plant. So it is relatively low and very well managed in most fields. So it's like very high prevalence, very high incidence, but because of the management in place, severity was low in most fields. So these numbers are more or less similar to last year, but just slightly higher. Last year was 90% of surveyed pea crops with the disease and 55% incidence and same severity. So slightly higher numbers compared to 2023. So moving to bacterial blight on field pea, symptoms consistent with bacterial blight were present in 35% of crops with average incidence of 7%. Both prevalence and incidence were slightly lower than last year. Bacterial streaming test was conducted with a microscope on representative samples from a portion of assessed fields. And for this disease, presence of this disease usually are seen to be influenced by crop damage due to adverse weather or insect damage. So plant injury will favor infection when the pathogen inoculum is present in the area. So one quite surprising event for us to see was very high amount of downy mildew in 2024. So we found it frequently in field pea crops, was present in 60% of fields with an average incidence of 23%, which were much higher than 17% of fields with an average incidence of 1.4% last year. So it was very frequently found and quite high incidence. Of course, it loves moisture, so it had the disease had the chance to align with a very favorable weather we had earlier spring. So it kind of took the ground, and many of those cases were reported from across the province. So root rot complex, a big issue for field pea, so was present in 90% of fields surveyed in 2024. Average incidence was 57%. Severity was 2.2, again on a 1 to 7 disease severity scale, with 1 as healthy, but overall a big issue. And it is like frequently high over multiple years. So last year was also present in 90% of fields, and average incidence was quite the same, slightly higher last year, 62%, and severity was also 2.5 last year. And that's the problem with this disease, because it's a complex and not all members of complex are necessarily love moisture, while the rest of the complex loves moisture. So it's the reason that it is frequently found in both wet and dry years. Only the composition of pathogens evolved will likely change. So again, as I mentioned, if you are interested to know more on regional breakdown of these diseases across province, all these reports are available online through Western Forum and Pest Management website under Western Committee on Plant Disease box. And this is like the table of regional breakdown for prevalence for field P diseases, and the other one is incidence of diseases across different regions for province. Again, similar pattern like lentil root rot complex and Mycosphaerella ascochyta complex were like evenly high across regions, while other diseases varied quite significantly from one region to another region in terms of their prevalence and incidence. So again, as I mentioned, this year we didn't have a soybean disease survey, but instead we had a drive-in disease survey for very first time in province because we thought this crop has lots of potential. So it's good to have some hands-on the plants and see what's going on, especially that we have more of the acreages under irrigation. So we selected 10 irrigated dry-bean fields, and we surveyed them in August 2024, between August 7th and 20th. And for each field, we assessed 10 plants for disease incidence and severity at 5 different sites, similar to lentil and field P, with a total of 50 plants per field. And again, we were careful that each site had at least 30 meters apart and located at least 50 meters from the field each. So each of the 50 plants were assessed for the presence of the following diseases: bacterial blight complex. It is very hard at the field level for a surveyor without a microscope in the field or without other tools to differentiate the three members of the bacterial blight complex, which are the bacterial brown spot and common bacterial blight and halo blight. So at the field level, we looked at them as a complex and recorded them that way. We also look at the Sclerotinia stem rot presence, anthracnose, and root rot complex. But when we sent the samples to the lab, we asked lab for each field to test if the field had any of those bacterial diseases like accurately. So the most prevalent disease, as we expected, was bacterial blight as a complex, and it was present in almost all fields. The average incidence was 66%, and the average severity was 0.7. For anthracnose, we found it in half of the fields surveyed. So in 5 out of the 10 fields surveyed, we had typical symptoms of anthracnose. We didn't find any Sclerotinia stem rot in dry-bean disease fields that we surveyed. And we were quite happy to see that we didn't see root rot issue as a big big like concern as it is for lentil and field pea for dry beans. We had just observation of 3 of the fields with root rot complex among the 10 fields, and incidence was quite low at 7%, and average severity was like very low at 1.1. Again, the same disease severity scale we used for field pea and lentil used here. So one is a healthy plant, so 1.1 is just slightly higher than that. So not a perhaps not a big issue as it is for lentil and field pea crops. Again, lab confirmed that 7 fields out of 10 had bacterial brown spot, and 6 fields out of 10 had common bacterial blight, and 3 out of 10 had halo blight. And if you add them up, they are more than 10 because they were fields that had more than one bacterial pathogen confirmed within the same field. So that was that was for pulse crops. So moving to fusarium head blight assessment that we do in province every year for cereals. I also will give you some high level results from that program. Looking at wheat first. So in 2024, we looked at 68 common wheat fields, which are Canadian Western Red Spring and Canadian Prairie Spring wheat classes, and 38 durum wheat, which are the Canadian Western Amber durum class. And the way that this survey is being conducted is Saskatchewan Crop Insurance Corporation crop adjusters will do the survey and randomly collect 50 sparks from crops at late milk to early dough stages. And then a subsample of 30 spikes kind of was analyzed for visual FHP symptoms and then for further analysis for speciation at the crop protection laboratory in Regina. So this is a lot of work to run this program. So again, 2024 FHB occurred in 28% of the common wheat and 18% of the durum wheat crops surveyed last summer. FHB was more prevalent in 2024 than in 2023 when it occurred in 24% of the common wheat and just 4% of durum wheat crops. So more prevalent definitely compared to last year. The provincial mean severity was 0.003% and 0.04% for common and durum wheat respectively. So about the pathogen species involved in causing those FHB in crops with FHB symptoms, Fusarium poae was the most prevalent species. It was confirmed in 84% of the common wheat fields and in all durum wheat fields, which is quite consistent with findings from last year. So because many people are also interested to know more about Fusarium graminearum because we are hearing about that one a lot. It's an aggressive FHB pathogen. It was also present in 12% of all wheat crops, was more prevalent in common wheat than in durum wheat, but was present. Other species were also identified among the FHB pathogen complex. A detailed report will be soon submitted to the Canadian Plant Disease Survey Journal. If you go to Western Foreign Pest Management website that I showed, you will see a preliminary report, but a detailed report will be submitted and the Canadian Plant Disease Survey Journal very shortly, and will be available later on to look at details of this program. So moving to barley. So in 2024, Fusarium head blight incidence and severity were also assessed in 51 barley crops in Saskatchewan by CIC and the Crop Protection Laboratory in Regina. These 51 fields was composed of 47 two-row and 4 6-row. And FHB occurred in 45% of the surveyed barley crops at a mean provincial severity of 0.93. The severity of FHB in 2024 was higher than in 2023, and that's because among the surveyed fields, there were fields that were heavily impacted by the disease. Looking at which species was the most prevalent one this year was Fusarium poae again in barley too, which was detected in 83% of barley crops with FHB symptoms. Again, all the methodology, everything was similar to what I explained for wheat. So the same staging and the same numbers and the same way of analysis. And perhaps I just come back to this slide again if we want to also hear something about the Fusarium graminearum. Fusarium graminearum was also present in 13% of the fields for barley too. So not as prevalent as POA, but still present here or there. So that was for the FHB program. Again, that's a huge program. I just summarized everything into a slide just for the interest of the time. But again, in addition to the preliminary report available already through the Western Forum on Pest Management, detailed report will also be available later through the Canadian Plant Disease Survey Journal, and all the reports from previous years, back to many years, are available on that website of the Canadian Plant Disease Survey Journal. So moving to comprehensive program, we also have for canola. This is quite a little bit not, yeah, it's quite significantly different because this is not one program. For canola, we have three separate programs. So for canola, we have general canola disease survey, and we have verticillium stripe specific survey, which is done after harvest, and we have perhaps one of the most comprehensive monitoring programs worldwide for clawroot, happens in Saskatchewan. So for general Canada disease survey, we looked at 208 fields in 2024. For verticillium stripe specific survey after harvest, we looked at half of those fields almost. So we selected half of those fields we did for general Canada disease survey, and we did the verticillium stripe specific survey on those, and we took to 103 fields in total, and clawroot monitoring program involved 482 fields. So this year, 208 canola fields were surveyed for the general Canada disease survey. During the survey, incidence and prevalence was recorded for diseases, and severity was rated for some of the diseases. For general Canada disease survey, we look at 100 plants in each field. So again, W-shaped pattern, 5 surveying site, but 20 plants per surveying site for a total of 100. Again, just to refresh minds, prevalence, the percent of crops surveyed that have the disease; incidence, the percent of plants surveyed within each field that have symptoms of the disease; and severity is the proportion of plant tissue that shows symptoms based on a certain disease severity scale. So I will check a little bit of what we saw for Sclerotinia stem rot during the survey. Sclerotinia stem rot was present in 56% of crops surveyed, at least at trace level. It was 30% last year, 56% in 2022. So it was higher this year than last year with 30%, but quite the same prevalence with 2022. So an average incidence, the average incidence across all survey fields was 9%, but in infected fields only it was 16%. And the overall severity was 0.24, and the severity in infected fields was 0.43 on a 0 to 5 scale. So here is a regional breakdown of Sclerotinia prevalence and incidence results from this year's survey in the province. Prevalence was highest in the northeast region, with almost all crops have at least trace level of disease, like at 97% or so. The disease was at lowest prevalence in the southwest regions. We saw it in 20% of the fields surveyed. Incidence was highest in the northeast region with 18%, and it was again lowest in the south. It was lowest in the southeast regions with 1%. So quite varied among different regions of the province. This graph shows and compares average incidence of Sclerotinia stem rot seen this year and compares it back to the previous years, dating back to 1999. And these are based on the historical data from the General Canada Disease Survey. This graph, I like this graph because it shows very well how dependent and reliable scrutiny is on environmental factors, particularly moisture. Again, if you look at this graph, we see that sclerotinia incidence peaked in 2016, which was a very wet year at almost 25% of plants with this disease. But right the year after, which was a very dry year in 2017, it dropped significantly to 3.4% incidence, and then it started to increase again and stayed at 17% in 22, then dramatically dropped in 2021, a very dry year, to record 2.2 low, but increased again in 2022 to only 6%, slightly declined to 4% in 2023, but now increased it to 9% this year, given the conducive environment we had. Again, this is a very interesting pattern for this particular disease in terms of its alignment with environmental condition. So blackleg is another very important disease. It is very important in terms of yield. It's very important sometimes for other reasons like trade. Blackleg is another disease that we look at it closely every year during this survey. Relatively has traditionally been less affected by environment relative to Sclerotinia stem rot. Uh, but this year we had like a perfect environment in the spring for blackleg, which led to like double the amount of blackleg we had in Saskatchewan compared to last year. So 92% of the crops surveyed in 2024 had at least trace levels of blackleg, and the incidence was on average at 23%, and severity was on average at 0.39 on a 0 to 5 disease severity scale. So almost I can say our numbers or amount of blackleg we got last year more than doubled compared to the previous year with an all-time high incidence. So here's a regional breakdown for incidence and prevalence of blackleg from this year. And as we see kind of across the province, it was very, very high. Uh, so uh, so overall, it was very, very high across the province. Perhaps southwest had a little had lower amount, but rather than southwest, across the province, we had quite significant amount of blackleg this year. So this graph is a comparison of blackleg incidence that was seen this year compared to previous years, dating back to 2011. Overall, there had been an increase in blackleg being seen between 2011 and this year. Incidence was high in 2020. If you look at that, 2020 was the highest before this year, still below 15%, at almost 15%. Then decreased in 2021 to 7%, then increased in 2023 and 2022 and 2023 to 11% and 12%. But now all of the sudden it's almost doubled to 23%, which I can call it an all-time high. So aster yellows, usually we consider blackleg and clawroot and sclerotinia and verticillium for bigs, but this is the second year we saw aster yellows very significant. So 25%, it was not as high as last year, but it's still very significant. So 25% of our fields had aster yellows. If we looked at the 100 plants addressed or assessed, so if we have like the 100 plants and aster yellows was between them and calculated prevalence that way, 25% of the fields had aster yellows, but plants with aster yellows are so evident in the field. So if a surveyor sees them even like far from the surveying site, they can also include in their form that they observed aster yellows, even though not within the 100 plants they assess randomly. If we add those trace levels, then 71% of the fields were seen to have aster yellows this year. But the number we should be worried is that 25%, because that can be linked to the to the yield later on. So incidence was 0.6% across all surveyed fields. And for incidence, it is just between those 100 plants assessed. So this number can be linked to the yield loss. And it was 3% in disease fields. So again, last year and this year, Aspergillus was not minor disease at all. It was a significant issue in certain field. It caused it's called significant damage. On the powdery mildew side, that we got really worried last year when we saw it in 13 of the surveyed fields picked within the survey, in addition to external reports. So last year it could be translated to 6% prevalence for a disease that is usually considered rare to happen across prairies. So we've been quite happy to not see it this year as common as last year. So we just saw it in 2 of the surveyed fields, which is good news compared to 13 last year. So moving to alternaria black spot, which we also assessed during the general canola disease survey, we looked at the pods and we saw the symptoms of alternaria black spot in almost 70% of the fields assessed. So that was it for general canola disease survey. So going to clover monitoring program in 2024. So the Saskatchewan Clover Monitoring Program is a collaboration, and it has its huge team. It's a collaboration among the ministry with SARM Plant Health Technical Advisors, or PHTAs, and Saskatchewan Crop Insurance Corporation and Sask Oilseeds. And to be successful in monitoring the spread and distribution of clover across the province and encouraging proactive on-farm management. Again, this year in 2024, we proposed it with 5 different components. It is a program 100% permission based, and with clover being done or being surveyed 100% permission based. Now, the whole pest survey program in Saskatchewan is 100% permission based. So these were the 5 components of the club route that I was talking about. The picture now you see, it is the way we did it traditionally in previous years. But as you may have heard or know, the disease, the club route, is not a declared pest or regulated pest in Saskatchewan any longer as of June 2024. So we modified the program. We kept the scope as it was in terms of its surveillance capabilities, but we kind of converted some of the more regulatory aspects to agronomic. One was following up on clover fields that we changed it to a client service. So still we asked for permission to get back to those fields, and it was interesting that many producers welcomed us back to those fields for free scouting and free assessment and free soil testing. Another aspect that had changed compared to previous years, reporting of the pest is when the pest is declared and regulated. So reporting aspect is not applicable anymore to clover root, but if a producer voluntarily wants to report it to us, we just reflect that in our map at the general location at rural municipality level only. So to have the maps updated, and we will help them whatever way we can in terms of testing and scouting and pathotyping, if anything found. But just that the changes didn't impact the scope of the monitoring program in 2024. We just did it almost the same way as previous years. We had 2 surveys. One was clover specific monitoring in high risk areas. And these are the fields with tight rotation, and we identify this information on the 4 out of 6 canola in last 6 years. So if a field has 4 years of canola in last 6 years, it's eligible to be surveyed under this program. The program is fully permission based. Of course, because these are tight rotation fields, these are considered quite high risk, but we also continue to monitor for club root within the regular general canal disease survey. That survey is being done by ministry staff, plant health technical advisors, survey volunteers from industry and agrologists. And club root is being assessed like any other diseases through the general canal disease survey. Again, we continue to offer support in managing the already known uh, infested club fields, but it was not mandatory. It was just permission based as well. With partnership with Sask Oilseed, we continue to have the soil testing bags available to producers and agrologists. The bags were available through the ministry regional offices by crop extension specialists and then also through the SARM PHTAs or plant health technical advisors, interested RMs, and so on and so forth. And then agrologists or producers could do the soil sampling and send it to Discovery Seed Labs for testing. Sask Oilseeds covered all the costs. Sask Oilseeds also covered the cost of testing for the crop root specific monitoring in high risks as well, in high risk areas as well. We continue to also raise awareness and continue to the extension work with publishing multiple articles. So we also had that as one component. All permission based, everything permission based. The only thing is the difference in methodology that for the general canal disease survey, again, as I mentioned a couple of times here, and for pulses, it is a randomly done survey. So we looked at the 5 surveying sites across the W-shaped pattern, looking at 20 plants in each field, and we look at for every disease, including club root, while for the club root risk-based survey, as the name suggests, it is a targeted, it's not random, it's targeted survey. So we are there to find a disease, kind of, I can explain it that way. So we looked at the areas that have higher chance of club root, like old or new entrances, low spots, water run, old yard site and stuff. And again, these are all permission based. Again, we had this fantastic program from Sask Oilseed last year in 2024 that everyone could test their soils for club root, kind of proactively. It's very important because if you find it in a very low level proactively in soil, then you can easily manage it without seeing a symptom. Overall, 181 fields were looked at as part of the infill surveillance in high-risk area under the risk-based portion, and then 208 fields through the general Canada disease survey. 34 fields we looked at among those that we have them as positive in our database with permission of the producers, and 59 soil samples were received directly submitted by producers and agrologists. So overall 482 fields were examined. We also published multiple articles, again continue to raise the awareness about club root because it is still a high risk disease. If we look at the if we look at it from the perspective of Canadian prairies rather than province only, it is still being frequently found on the Canadian prairies, and it's good to be very careful about it. So this small table actually reflects the whole history of club root in canola in the province and shows how the numbers accumulated to a total of what we see in the table. So this year and last year, we have not seen any club root symptoms within the club root monitoring program with almost 500 fields that we looked at. So it was great news both this year and last year. This year we found 2 additional fields with pathogen DNA found in the soil, but without any symptoms. So just the pathogen DNA were found in the soil and producers were contacted. So they are in a good shape to manage the disease without letting the symptoms develop. And because of that, 2 new fields were in a new RM. So 1 new RM was added into our map. So, and that was RM439, which was not a surprise to me because it was already surrounded by RMs with reports of clawroot visible symptoms. So again, every year, the general locations of all clawroot infested fields to date, we will use it to update the Saskatchewan clawroot distribution map. And this map is only at the rural municipality level. And it is just to raise awareness that where we saw the disease. Again, this is a cumulative map with the results from 2008 to 2024. But specific to this year, through the climate monitoring program, only one new RM was added to this map, and only two new fields were found to be infested by clawroot. So again, we didn't have any sample this year to send for pathotyping. Usually, University of Alberta supports us for pathotyping, but we didn't have any to send this year. We didn't have any to send from last year, so we haven't had anything to do pathotyping for. But in the past, every year we send samples to the U of A, and University of Alberta did all the pathotyping for us, and we have never had any path types that are known to be able to break resistance in Saskatchewan on a commercial setting, on a commercial line. But still, we should be very careful, as aggressive races in Saskatchewan were reported through the racing program from AFC in Saskatoon. Um, so it's important to just be cautious about clawroot, and uh, it was a very successful. It is a success story for Saskatchewan. Not seeing it for 2 years and with visible symptoms is great, but still we should continue doing our due diligence against this disease. So, Clubroot's management plan for Saskatchewan gets updated every year by the Saskatchewan Clubroot Committee, actually, which it meets on February 10, I guess, or 13 this year. So very, very shortly, it meets and updates the Clubroot management plan, but also clubroot.ca through the Canada Council Canada is a great resource with almost any information needed can be found there. Of course, that's for all the canola diseases, including clawroot. Moving to another disease that has its own monitoring program, it's Verticillium stripe in canola. So, like clawroot, Verticillium stripe has now, at least currently, has its own program with multiple components, and just this is quite a recently uh, reported disease from Western Canada in general. The first time it was discovered in Manitoba, and not long time ago, just in 2014, and then in 2015, CFIA kind of did like a across Canada testing, and the DNA of the pathogen was found in Saskatchewan too. Uh, so it was somewhere for sure that the DNA was found, but never officially reported and confirmed in Saskatchewan with visible signs of the pathogen in commercial fields until 2021. So everything for Saskatchewan is new. For Western Canada is almost new. So we have lots of things that still we don't know much about when it gets to Verticillium stripe. But a lot of research are going on in multiple research labs, and we are hoping to receive more and more information on the Canadian context. So, yeah, I put this picture because it is actually the very first commercial fields with visible signs of the pathogen reported from east of the province by a producer who is also a plant health technical advisor. And it was in 2021. So we had that report, and then uh, we thought that the disease perhaps is not as rare than everyone was thinking. So we had like a pilot project in 2022. We went to far east of the province with the whole group. Each of those color on the east side is representing one surveyor that they did the survey in, I can say, late September, early October. We sent samples to the University of Manitoba for supporting in terms of lab analysis. We got lots of lessons from that small pilot project. We had 15 of those 25 fields with suspect samples, but quite out of 15, 7 were confirmed as verticillium, but only 3 of the 15 confirmed as verticillium longisporium. So we learned not all those plants with suspect or suggesting symptoms will got back confirmed from the lab. And on the other hand, we also found that other species of verticillium may be involved in that disease beside verticillium longisporium. So now that we got those information last year and this year, we decided to have like a 2 years project kind of survey and just survey half of the fields that we do for general cannula disease surveys, select half of those and get back to them later in September, early October, uh, and just specifically look for verticillium and collect samples and send to the lab for analysis. So the map you're seeing is from last year and not from this year, and the positive RMs are RMs that were found positive through our program or through the Sask OSIS program. So if a producer sends sample privately to a lab, it is not reflected here. So you can expect the number of RMs was more than what we can see on this map from last year. But we learned last year something new, that the disease, even though it was mainly found on the east side of the province, but we had it like look at that RM111 just as far west as which as far west as uh, the area actually bordered with Alberta. So it was found even last year across the province. So we learned that the disease is not as rare. It is perhaps more spread than initially thought. Moving to this year as the second year of the program as the whole scope after harvest verticillium specific survey, we did it again, as I mentioned, on 103 fields across the province. Half of those fields we did for general Canada disease survey. Of course, the lab portion was done in October, and the lab analysis was just being finalized now, and or actually have just been finalized, and now we started to contact producers that kind of got impacted by those findings. And later on, when we are sure our producers are aware of their field level result for verticillium stripe, we are gonna put together similar map for 2024. But this year we also going to put together an accumulative map, like what I showed you for clubroot. So one for just this year and another one cumulative, and we are trying to include as many reports as we have access to, like from 2021 and beyond, and see what it looks like when we look at the cumulative map in terms of the spread of the disease. But definitely, verticillium stripes no longer a rare disease in Saskatchewan. I can tell you that just based on the rough calculation we did on the prevalence of the disease this year, 25 to 30% of surveyed fields were confirmed with verticillium stripe by lab. So this is quite a huge number. On the other hand, we've heard anecdotally that 2 to 3% of the fields from Alberta may also have the disease confirmed. So it seems now it's not Manitoba disease, but it is across the prairies. Again, lots of great research are being done by researchers and by industry, and we are looking forward to like more solid data and information from researchers for every aspect that can help producers in management of the disease, from especially from the resistance development side and resistance characterization of the genes involved and everything. Again, lots of good research are going down, and some of the research summaries are available through through Canada Council of Canada website already. Yeah, this was just my very first field this year that I personally looked at, and it was already positive. So if you look at that single piece with the lines around it, you will see a ton of macrosclerosia of verticillium stripe already present under the peeled back epidermis of the stem, which is a confirmation so for this disease. So, and again, now that we have the lab results, we will see 25 to 30% of the fields assessed this year got confirmation from lab with positive verticillium stripe presence. I assume this was my last slide. So again, this is a huge work done by many people. I presented it on behalf of this huge team. Uh, so if you have any question, you can see three names on the slide. So myself as a plant disease specialist, but most surveys, almost all surveys, gets coordinated by Carter Prue, our provincial specialist integrated pest management, while a cloud root monitoring program is getting coordinated or has been coordinated by our provincial specialist pest regulatory, who is Teo Adigeye. So if you have any questions specific to cloud root monitoring program, you can reach out to Tayo. And if you have any question on other surveys, you can reach out to me or Carter Prue. So that was it. Thank you so much for your attention and for joining the webinar today. And thanks uh, the ministry to invite us to share these findings.
Thank you, Alireza. That was a great presentation. We have a bit of time for questions. I just want to remind everybody to stick around until after the question period for the CCA credit QR code. And anyone with any questions, please type them in the question box. So yeah, I think we have quite a few questions around blacklegs, since there was such an uptick this year. What environmental conditions lead to the increase in blackleg, and what should producers be doing to manage it?
So again, blackleg is one of the diseases that has quite a known pathosystem and lots of management tools when we compare it to verticillium, with almost very limited amount of tools in the management toolbox. Blackleg is in a good shape, but perhaps I will divide the question to two parts: one on environment, or why we saw this like raise in amount of blackleg. Quite for incidence, it is all-time high overall, twice compared to last year. We analyzed the environmental condition we had very early spring, and that is the only reason. So there is no more reason. There is nothing related, because it is last year versus this year. So it is nothing about cultivars or practices or anything. It is just the environment was perfect for the like I can say strong establishment of the disease in very early spring, with lots of shower and most moisture at that timing. So it helped the pathogen very well settle. So at that time, we observed and we got anecdotal reports from across the province that people reported leaf lesions caused by blackleg, with those typical like pycnidia in a concentric form that is quite specific to blackleg. So the inoculum was there. Excuse me. In terms of management, again, this is a rich pathosystem in terms of its management, with many tools available. So quite advanced advanced system for resistance. Almost half of the seed companies now have the resistance grouping on their labels, in addition to general resistance categorization that was in place for many years, like resistance versus moderate to susceptible and stuff like that. It was there for years, but now more than half of the products have also the exact resistance group, like E1, like A, B, things like that. And with the blackleg race testing available through the labs, and of course it was available through Sask Oilseeds for free in 2024, as was in the previous years. Uh, then you can sense like stems, and then they can tell you what type of pathogen race profile you will see in your samples, and then from there you can find the resistance genes that you want in your host, which is your canola plants. And then from there you can find out which resistant group you need to grow next time that you plan to grow canola, if blackleg is a limiting factor there. And then there's a nice table on the Canola Council of Canada website that has those grouping, that, like, from, for example, this certain resistant group, we have these cultivars from these companies available. So you can select the next cultivar that you want to grow in your field, like with a lot more information compared to the past, with these testings for pathogen race profiling available through multiple labs now, and through Sask Oilseed, at least for 2024 and before. And I assume they will continue that very nice program, but I cannot talk on their behalf, but it's my understanding. But again, the testing is available, so you can go do the testing, find out the pathogen profile, find out the genes that you need, find out the resistance grouping that you want to see on the label, and then find out the exact cultivar that you want to grow. And if you need more information, all of these are very well laid out on the Canola Council of Canada website under how to interpret blackleg race testing. So that is for the resistance side of stuff, very well developed. So you have lots of option to grow if blackleg is a limiting factor. If you want to start looking at blackleg, because it's different for every field. One field has no blackleg, but like has shattering. So that's the more important trait to start from. But if the blackleg was a limiting factor, then you can start to choose your next cultivar based on blackleg, very well informed. For the seed treatment side of the stuff, there are now good seed treatment options available. All of them are mentioned in the guide to crop protection, and the new guide to crop protection will be available in spring. But there are multiple options now for seed treatment against blackleg, and even some of them have the airborne inoculum now on the label. Most of them have the seedborne on the label, but now some of them even have the airborne blackleg on the label, which is good. And if there are all the risk factors in place, and like you had lots of blackleg last year, and you think that you have a very heavy percent of inoculum in your field, and you want to do a spraying, even though it's not common in Saskatchewan, but there were fields that is sprayed against blackleg this year at the seedling stage, and there are multiple products available to do that spraying. So there are a lot of tools available for the management of blackleg these years.
No, that's a really good summary of the tools. I just want to mention again, if anyone missed, that the QR code for the CCA credit will be available after the question period here. I've got quite a few questions, Alireza, so we'll just try to flip through them quickly if we can. Similar to the theme, um, do you see any of the resistance of the canola breaking down? Would that have been any reason why we've seen higher incidence this year?
Yeah, you mean for blackleg?
For blackleg, yes.
Yeah, I don't think so. I don't think so, because because it is a comparison of just what happened this year to last year, and the main changes that can happen in such a short term in terms of time window is can just be related back to the environment. So I don't think any significant resistance breaking happened specific to this event or raise of black like we observed. So briefly,
That's good.
yeah.
That's good to know. And you mentioned crop rotations can be a good tool, but another person asked if intercropping can be a good tool for blackleg.
So that's because the multiple fact. Of course, rotation is great because if we can get away for 2 years from canola, so a 2 years break between the two canola crops is a really nice time to for those Pseudothecia or Pycnidia. These are the fruiting bodies of the pathogen to be decomposed. But intercropping, if no, of course, if yeah, so it's almost it's a complex scenario on what we are intercropping to see what would be the impact on inoculum. But if our intercropping crops include one brassica plant like canola or mustard, then perhaps it is not a very useful practice in terms of lowering the amount of inoculum in fields for blackleg.
Interesting. Next question we have is, was there any evidence of individual canola plants being infected with multiple diseases this last year?
Yeah, that's very common. So it is not rare to see one plant with multiple diseases. I personally saw a lot of stems that they had verticillium stripe and blackleg together. I guess it is frequently found by almost all surveyors to have one stem, and single stem has both blackleg and verticillium. Also, most of those stems also have gray stems, so I can say it is common to see blackleg and verticillium and gray stem together. But we also had stems that they also had sclerotinia on the same plant that had verticillium and blackleg and gray stem. So yes.
So would you say that blackleg and verticillium share the same environmental conditions for development and infection, or what is your comments on that?
So, Sclerotinia belongs to a set of pathogens that they are very heavily dependent on environment. So their occurrence patterns are very well aligned with environment. Blackleg, however, is still loves wet and most kind of wet and warm weather in early spring, but it is traditionally get less impacted by environment. So even in a dry year, you will frequently find blackleg. But there were dry years that finding a field with Sclerotinia was quite very rare because of the drought, but it is not the case for blackleg. Blackleg is a resistant pathogen, so it is quite different from Sclerotinia, I can say, to address the question, and its needs are quite different. It's more resilient to the environmental pattern compared to Sclerotinia that is heavily dependent.
Sorry, Alireza, I think I may have misspoke there, but the person was asking verticillium and blackleg. I think I said sclerotinia the first time, but
Or I heard, yeah, perhaps either of us made mistake. So that's fine. So verticillium versus blackleg, quite different. Again, quite different. Even though that seeing them both together is not rare. It's quite common to see both verticillium and blackleg, but their needs are a little bit different. Their pathosystems are a little bit different. So blackleg again likes at least at this certain time like not likes like wet and warm moisture. Verticillium, however, is living within the xylem. So it likes moisture just to help itself to germinate as a macrosclerotia and get to the xylem. After that, it loves hot and dry weather or anything that further stresses the plant and lowers the amount of stream in the xylem. So verticillium later on in the season prefers hot and dry compared to blackleg.
Okay, and another question coming in here. In regards to aster yellows, did you see any canola hybrids in particular that had disease present more than any other?
So that's a hard question to address, but I can say that the fields that we saw with more incidence of aster yellows were more conveniently linked to the seeding date compared to any specific cultivar like with more or less aster yellows. Aster yellows is such a complicated disease. It's a vector-borne related disease, and it is very well linked with infected leaf hoppers coming to our area. And Dr. Tyler West in AFC Saskatoon does have a very great program looking at different aspects of this very complex pathosystem, and they found out lots of great results. But this is a very complex disease, mainly related to the amount of actually as leaf hoppers that you get with aster yellows within them, and also perhaps some impact of seeding dates on this disease. But I personally don't have any information on on actually linking certain cultivars versus the amount of the disease observed.
Yeah, I know that's a tough one. It kind of seems to be all over the place. I've got
Yeah,
one last question for
yeah,
you.
but before we wrap up on that question, you can I guess you can ask this same question from Tyler West in AFC Saskatoon, and then you receive like way more detailed information.
Okay, okay, I got one more quick question here. How far away are we from either resistant cultivars or a treatment regime for verticillium stripe?
So this is a very good question, frequently asked. With psyllium now spread, now everyone is trying to enrich the management toolbox to handle this disease. So lots of good research is being done to characterize the genes in cultivars for verticillium stripe. There are some progress on that, and one of the great programs is Dr. Hussein Borham program in AFC Saskatoon. But other studies that they are looking at resistance screening are uh, also trying hard across the prairies at the U of M, at the U of A, and other research labs, and also the industry is doing a lot. So there are two aspects when we talk about the resistance. One is resistance characterization, like knowing exactly what gene or genes are involved. And another aspect is just the normal general screening, so like resistance screening against the pathogen. So from the first aspect, which is the resistance characterization, research works excuse me, are continuing and in progress, especially in those programs that I mentioned. But both those programs and industry are doing a huge amount of resistance screening, and they found out that uh, there are significant differences in the level of reactions exhibited by different cultivars, and they are working to understand the background. But just just personally, and my personal thoughts are, we are still away a little bit for perhaps couple years to see the first cultivar with like a characterized gene in it. But on the other hand, already some of the companies started to put verticillium stripe rating on their products. So there are companies that now you can see that they already put, based on the resistance screening, they put the level of the reaction or verticillium stripe rating on their product that can be used. Uh, for the fungicide aspect, I am also aware that that work started with some companies, so we may be able to see some products in future, but nothing that comes like in a short term. So
No,
very
that's
brief, yeah, very briefly now we are quite relied on rotation to manage this disease.
no, that's good to know. Um, we're just about out of time here, so thank you